Hello to everyone. We're gonna start to record. Um, it's nice to be here this Friday again for another Solanaceae seminar online. Before to start with the talk, I had the honor to introduce to Natalia Pabón Mora, who is going to present the talk entitled the Anatomical and Genetic Basis of Fruit Diversity in Solanaceae. Well, I have a short biographical sketch here from Natalia. So Natalia is an associate professor at the Universidad of Antioquía in Medellín, Colombia. She holds a PhD from the joint program between the CTU University of New York and the New York Botanical Garden. And she's leading researcher of the Plant Evo group, focused on the study of the tropical non-model plant species. In 2015, she was awarded with the Early Career Award by the Pan American Society for Evolutionary and Developmental Biology. More recently, in 2019, she was recognized with the GWET Award from the Arnold Arboretum at Harvard University. And she's currently the Dresden Junior, had a Dresden Junior Fellowship at the Technical University in Dresden, has a visiting professor there. Thank you, Natalia, to accept this invitation. And thank you to everyone to be here today. Natalia, can please share your screen and start with your presentation? Thank you so much.
Andres, thank you so much for the introduction. Thank you, and thanks, Rocio, for this invitation. I'm gonna start sharing my screen now. I'm gonna go ahead, and if there is anything wrong, you guys can stop me and tell me. Okay. So I think everyone is seeing right now the first slide of the presentation. And like Andres said, today I'll be here with you sharing our research on the anatomical and genetic basis of fruit diversity, specifically in the Solanaceae. So just for you to have an idea of what we do in the group, like Andres mentioned, I am the principal investigator of the Plant Evo group at the Universidad de Antioquia. And we're mainly interested in understanding the genetic mechanisms underlying flower and fruit development and diversity. And we are interested in those two features, particularly because these two features are the keys in apomorphism for angiosperm diversification. And so perhaps they have aided in the radiation of angiosperms, as you can see here by the comparison in terms of the number of species. Angiosperms have reached around 3,000 species, 300,000 species, when compared to gymnosperms, which are only around 1,500. So in the group, we have at least 4 different research areas. I just want to tell you a little bit about what we do. We are interested in understanding the genetic mechanisms underlying floral organ identity and floral symmetry. We take as a starting point, as a reference point, the model organisms Arabidopsis and Antirhinum. We take those genetic models and we try to understand if the same genes do the same processes in other plant species that are perhaps more diverse. So we are interested in studying Orchidaceae, Tropiolaceae, and Aristolechiaceae, among other families. We're also interested in understanding flowering time in non-model species. And so for that, we have been looking into plants with huge ornamental potential, like the Orchidaceae family, that have long or extensive vegetative phases. So that's something that we're also interested in understanding and perhaps manipulating in the future. The other research area that we have is extreme development in parasitic plants. So we currently are interested in understanding this relationship between these two plants. What you see in the back is the host plant, that's Dalea quatrecasaceae, is a member of the Fabaceae. And what you see in purple are the sprouting floral buds of Pilostyles boyacensis, which is an apolantaceae. So that's a very extreme relationship between those two plants. And what we're mostly interested in understanding, how is it that the apolantaceae floral buds develop so quickly from parenchymatic pockets into quickly develop floral buds and then fruits. Finally, we're also interested in understanding the anatomical and genetic basis of fruit diversity. And for that, we have been looking into very many different plant families, including the Solanaceae, but we have also studied a little bit the Rubiaceae, Papaver and Chloranthaceae. Okay, so why studying fruits? So it turns out that fruits are extreme ontogenetic, are the result of extreme ontogenetic changes of the ovary wall after ovule fertilization. So you can see how extreme that journey can be over here. This is an example using Brunfelsia australis. And you can see how these are the different developmental stages of the flower, and then transitioning into the young fruits and the middle stages of the fruit development until that vessel is properly formed and is willing or able to release the seeds. Now, something that is very important is that the carpels start out in the center of the flower as very typical leaf-like organs. They don't present a lot of modifications early on, but they quickly specialize during mid-development into three portions: the stigma, the style, and the ovary. And the ovary is the one that holds the ovules, and it's really only after ovule fertilization that this transition starts to occur. So this transition is accompanied by both morphological changes, but also, and perhaps more importantly, anatomical changes that are related to the mechanisms that will result in seed dispersal in different fruits. Now, because fruits are at the key of plant reproduction, clearly studying their features is something that is important. Okay, the other thing that I think is important to highlight here is that seeds are older than fruits. So that's perhaps very obvious for these audience, but it's important to think that seeds are in fact older. They evolved around 280 million years ago, and it's really only with the emergence of angiosperms that we see all this diversity derived from the ovary, but particularly the transitional stages from the ovary to the fruit. And this is important because fruits may have aided also to enrich the diet of modern birds and mammals specifically, including ourselves. So you can see a little bit of that diversity, a snapshot of that diversity in this slide. Now, also something important to highlight here is that because of this diversity, fruits have been classified in several different manners and ways. But perhaps the two major categories for fruit identification or for fruit of classification are the dry dehiscent fruits and the fleshy fruits. And usually we can think of these fruits as the two most distant categories because in the dry fruits, we can always see thin pericarps, that's the fruit wall, usually accompanied by the lignification of the inner layers, which we call the endocarp, and the occurrence of dehiscent zones for seed release. Conversely, the fleshy fruits are very different. They develop a lot more layers, so there is an increase in cell division, usually an increase in cell expansion, and the ripening process is accompanied by accumulation of aromas, volatiles, and deliciousness. So two very different processes in order to distribute those seeds. Now, perhaps the most interesting fruit categories are those that don't fit in either of those two previous categories. So there are also the drupes over here. Those are the ones that have the potential to develop layers that are fleshy in the outside and different layers that are hard in the inside. Aggregate fruits, where many carpels come together into a different, into a joint structure. The multiple fruits where not only carpels form the same flower, but in fact different flowers come together to form the fruit, and they can even bring in some additional structures like the bracts or the inflorescence axis. And perhaps very interesting as well are the pseudofruits, which are the ones where fleshiness is derived from structures that are not the ovary wall. So for example, the receptacle in a strawberry, or a case that we have been studying, which is those fleshy bracts that occur in the genus Ediosmum in the family Chloranthaceae. Okay, so why the Solanaceae? So today I'm here to tell you, I have organized this talk so I can tell you everything that we have done in terms of trying to understand both the anatomical basis and the genetic basis for fruit diversity in the Solanaceae. And as it turns out, the Solanaceae is a wonderful framework to study fruit development because most of the, I know a lot of people don't like this word, but most of the early diverging taxa have dry dehiscent fruits. And then only in the diversification of the Solanaceae, we can see that really is the fleshy fruited species, which are the ones that are most abundant. So fleshy fruits are a synapomorphy for the subfamily Solanoideae. That's where most of the things that we eat are located. So they include, of course, you know, peppers, tomatoes, gooseberries, aubergines, among others. Now, interestingly, not everyone got the memo on time, so there are some independent acquisitions of different fruit types in the Solanaceae. So, for example, independent acquisition of fleshy fruits have occurred in Cestrum and as well as in Duboisia, and reversals to dry dehiscent fruits have occurred in the tribe Datureae, which include the genus Datureae and the genus Brugmansia as well. So you can see how in this framework, where it's mostly consistent that there is a single shift, some reversals have occurred. Now, interestingly, if we look at the very young developmental stages for carpel development, it's impossible to predict which one will be what type of fruit. The same happens with the teenage ovaries. So those pre-antheric flowers that have not started fruit development yet, you can't predict how the fruit will be. So basically these ovary walls are formed by several parenchymatic layers where it will be impossible to tell which one will become a particular fruit type. Okay, so the first thing we wanted to assess, and this is a work that started almost 10 years ago, was to really understand what were the main anatomical shifts that occur during fruit transition in different Solanaceae species. So in order to do so, we selected a few species having different fruit types in representative developmental stages. We performed standard anatomical analysis for most of these species, and we recorded specifically those spatiotemporal transformations occurring in the fruit wall, trying to compare also the location of those shifts. So we were very carefully looking at the apex, the mid portion of the fruit, as well as the base. And then finally, the whole goal here was to compare the features between the dry and the fleshy fruits. Okay, so I'm gonna walk you through the main results in terms of the anatomical data. We have been sampling over the years different taxa. And so I will start with the early diverging dry dehiscent fruits, and some of the taxa that we sampled include Brunfelsia, Cestrum, Nicotiana, and Petunia. Okay, so I'm going to start with the typical features that one can find in a dry dehiscent fruit in the Solanaceae. Here we're looking at the cross sections of Nicotiana sylvestris. You can see that I have labeled for you the apex, the middle portion of the fruit, and the base. And we had followed the developmental series at 3 days post-anthesis, 4 days post-anthesis, 5 days post-anthesis, and 7 to 8 days post-anthesis. And basically, I'm not going to tell you all of the changes, but I want to summarize what we have seen in these 6 features. So basically, every time we look at the cross-section of a dry dehiscent fruit, we see a constant number of cell layers. This number of cell layers doesn't change too much between the ovary wall and the fruit, the mature fruit. We see a sclerified endocarp, which is not unusual. Like I described before, this is a very common feature for dry dehiscent fruits. You can see how it starts over here at 5 days post-anthesis. The formation of lacuna, that's the intercellular spaces between cells in the pericarp. You can see those with those black arrows that I have for you over here. The lignification actually has a direction. So it starts centripetally and basipetally. It starts from the apex to the base, but it also goes from the outer layers of the endocarp to the inner layers of the endocarp. There is the formation of the dehiscent zones. Those are formed by the discontinuities of that sclerified endocarp. And because of those areas that are not lignified, the fruits can either dehisce septicidally or localicidally. So there can be septicidal or localicidal dehiscence, or both in different fruit types, different capsules. Okay, so we did this for a number of species. No? So Petunia, for example, has the same features. I'm not going to tell you everything again, but you can see how basically all of those features that I just mentioned before are occurring also in the dry fruits of Petunia. So those turn out to be common features for dry dehiscent fruits as well. What happens if the capsule is a little bit bigger, like in the case of Brunfelsia? So it's basically the same, although there is a little bit of an increase in the number of cell layers. All of the other features are retained. So basically, same dehiscent zones are still present. Lignification occurs in the same direction.
Oh, sorry, Natalia, just trying to let someone in. We muted. I think.
I think,
Yeah.
am I unmuted? Am I good?
Yeah, now it's good. Sorry.
Okay, no, I'm sorry. Okay, so what about the cestrum fruits? No, these, as you may recall, these are fruits that are fleshy, but are phylogenetically more related to the dry dehiscent fruits. And so here, something very interesting happens. You can see here, again, the apex, the middle portion of the fruit, the base of the fruit in different stages, 1.5, 2, 2.5, and 5 millimeters. And we still see some of these common features. So we can see a constant number of cell layers. We can see the formation of a sclerified endocarp, which is something that we wouldn't imagine if we only see the fruit from the outside. We can see the formation of lacuna, those intercellular spaces, and we can see that lignification follows the exact same pattern, centripetally, occurring centripetally and basipetally as well. So those features are very common in the dry dehiscent fruits. Oh, wait, something, okay. But there are also some lost features in the cestrum fruits. So for example, the dehiscent zones, those discontinuities of the sclerified endocarp are not so obvious, and they do not lead to the dehiscence, to the septicidal or loculicidal dehiscence. Although there had been reports for at least 2 different cestrum species that do dehisce in the apex. Now, there are also some new features in these fruits. So for example, we can see the expansion of the pericarp cells, and we can see processes associated with ripening in those particular fruit stages that are the older stages. Now, we were curious because this is a work that, as I already mentioned, I started a long time ago at NYBG. We were always curious to know if this was a feature that we had only recorded or seen in one species of cestrum. And so more recently, Natalia Hernandez-Ciro, who recently graduated from her undergraduate in biology at the Universidad of Antioquia, she repeated the same sections for a different species of Cestrum nocturnum, and she was able to identify the same features. So you can see that the lignified endocarp is not as dramatic as in Cestrum diurnum, but you can see the same common features, the same lost features, and the same new features as in the other species of Cestrum. Okay, so I want to move to the Solanoideae. So this is the next clade. This is where all the species have, or most species have fleshy fruits, except in this case for Brugmansia and Datureae. Okay, so what are the common features in the fleshy fruits? Very different features are seen here. As in comparison to the dry dehiscent fruits, we can see an increase in the number of cell layers. We can see that the fruit does growth, and this growth is due to both cell division, but also cell expansion. There is, in several of these species, there is presence of collenchyma, perhaps to give it some strength to the tissue. There is expansion of the endocarp into the locules. So that's something that is slightly different compared to the capsules. And of course, the final stage for the fruit development in these species is accompanied by ripening. Now, some of the lost features when compared to the early diverging dry dehiscent fruited taxa are the lack of that sclerenchymatic endocarp. You don't see anything red here except for the cuticle, but that's a very different structure, and the lack of adhesions. So this is the same for a lot of the species, even Iochroma, which is again a fleshy fruited species. It has the same common features. It has the same lost features as I mentioned before, but it also has a unique feature. We have not recorded this. It has been reported in the literature, but we only saw this in the fruit of Iochroma, and that is the presence of these sclerenchymatic bodies in the pericarp. These are the seeds. These are not the seeds. Those are called the sclerosomes. So basically, those are associations of sclerenchymatic cells that form little stones in the pericarp. Okay, let's look at the reversals, the independent acquisitions of dry dehiscent fruits. So here there is something that I think is fascinating because they keep a lot of the common features for the fleshy fruited species. So you can see that it doesn't matter if we're looking at Datureae, I will show you Brugmansia in a second. All the common features for the fleshy fruits are retained in this taxa. Which ones are those? They represent the increase in the number of cell layers, the fruit growth due to cell expansion. There is also presence of collenchyma. You can see it outside of the fiber or fibers that are pointing to the spines in Datureae. There is the expansion of the endocarp. There is the ripening. Same loss features and some new features. So for example, in the case of Datureae, we can see these fibers associated with the spiny outgrowths, and we can see occasional irregular dehiscence in these fruits. Something that I think is more striking in Brugmansia, but that it also occurs in Datureae, is the fact that the vascular tissue develop something different around it. And so there is degradation of the pericarp around the vascular tissue, which might help the dehiscence of these fruits. Like I said before, it's the same for Brugmansia suaveolens. We can see here, this was also done by Natalie last year. Some of the common features are retained, some of the loss features, or all of the loss features are retained. And the new features that we were able to record for Brugmansia suaveolens include that pericarp degradation that I was telling you about, lacuna around the vascular tissue. We can see that over here, especially in these figures, where there is an important accumulation of intercellular spaces around the vasculature. And there is also something really interesting, and that is the changes in cell orientation between the outer layers of the pericarp and the innermost layers of the pericarp. Perhaps aiding in different processes in late developmental fruit stages. Okay. So how can we summarize these results? You know, this is all really interesting. I love the anatomy of fruits, but what does that really tells us in terms of the evolution of fruits in the Solanaceae? And I think this really points to two important critical features that we need to consider. The first one is that it seems that the anatomical features are phylogenetically fixed. So it doesn't matter if in the early diverging taxa we're seeing dry dehiscent fruits or fleshy fruits. What matters is that we're seeing a lot of phylogenetically fixed anatomical features. No? So these include the retention of the same number of layers, the initiation of the endocarp that is lignified, among others. Now, the same is true for the Solanoideae. So it doesn't matter if these fruits are going to be fleshy and will ripen, or if these fruits are going to dehisce and will dry open and dehisce. We are still seeing some anatomical features that are phylogenetically fixed, like the increase in the number of cell layers in the pericarp, the cell expansion, and the accumulation of a cuticle. So what this tells us is that really fruit type is a late ontogenetic feature. No? So it might be that fruit type, what we're calling the fleshy fruits or the berries versus the dry dehiscent fruits or the capsules, might rely perhaps in a few genetic changes that occur at the end or during mid stages or during late stages of development. Okay, so I'm going to move now to the genetic basis of fruit development, because that what I just showed you has to do with the anatomical basis, but we have also studied some of the genes that we think are related with these changes. So the genetic basis of fruit patterning and dehiscence have been best identified in the model species Arabidopsis thaliana. Here you can see how in Arabidopsis there are two carpels that form the valves. The valves meet in a middle partition, which we call the rheplum. Now, during fruit development, those valves separate and leave the seeds attached to that middle partition until the seeds are shaken away or shaken off. Now, this is how it looks anatomically speaking. There are the two valves over here, and they are joined in that middle partition that we call the rheplum. Here you can see how during development, those two valves separate from that middle partition, and they do so by or through the dehiscent zones, which are the tissue layers that will break through in order to release those seeds. Okay, so genetically speaking, I want to make this as simple as possible. There is a first genetic regulatory tier that includes 3 transcription factors: fruitful, rheplumless, and shatterproof. Fruitful is key to control cell division in the fruit wall or the valves. In fact, you can see that in the fruitful mutant, the seeds just keep regular growth, but the fruits cannot keep up with this growth because they don't have enough cell division rates. And then the fruit basically prematurely ruptures and release the seeds prematurely. Replumless, on the other hand, is important to form, or it's important in the formation or the specification of this middle partition, which we call the replum. And then shatterproof is key to produce those tissues that will differentiate the dehiscent zone. So basically, shatterproof is a key to form those two layers of tissue that will form that area through which the pericarp will break through in order to release the seeds. You can see that in the double mutant, shatterproof 1 and 2, those layers are never formed and the seeds are never released. Okay, shatterproof. And in the second regulatory tier, shatterproof activates 3 more genes: INDEHISCENT, ALCATRAZ, and spatula. Now, something that you can think these names, INDEHISCENT, ALCATRAZ, and spatula, are all kind of reminiscent of enclosure of the seeds, fruits that are unable to release the seeds. And this is because those genes control specifically the fate of those cells that will form the dehiscent zone. So for example, INDEHISCENT is important to form, or in the formation of that dry sclerified layer, whereas all the other genes, including INDEHISCENT as well, are important for the formation of that layer that will never undergo lignification, through which the pericarp will break through. Finally, we have APERALA2. APERALA2 is a gene that negatively regulates the entire fruit genetic network. Now, something that is perhaps very interesting for you, if you recognize these gene families, is that these genes are members of large gene families, including the Matchbox genes, the homeobox genes, the BHLH, and the AP2ERF transcription factor families. So these are genes that are very diverse in flowering plants. Okay, so because our study systems are usually not model species, we start out by generating reference transcriptomes for those taxa that we're interested in studying that don't have a genome or a reference transcriptome. So we start over here, we collect the stages, as many developmental stages as possible in the field, including floral buds, flowers in pre-anthesis, and young fruits, and leaves from different taxa. So in the Solanaceae, we have concentrated our attention on these 4 taxa: Streptozolen hamiltoni, Brunfelsia australis, and the reversals. We were very interested in the fruit reversals, so we sequenced Cestrum nocturnum and Brugmansia suaveolens as well. Once we have those reference transcriptomes, we do a second step or an important step, which is the search for homologs. So in this case, we're looking for all of those genes that are important in fruit patterning, taken as a reference model, the Arabidopsis genetic. Now we move, once we have found or identified those homologs, we move into the phylogenetic analysis. Basically, this is done to confirm the homology of those genes and to map for duplication events. This is something that I will discuss a little bit later. And then we move to expression analysis. So in this case, what we have done is we have incorporated RT-PCR, real-time PCR, or quantitative real-time PCR, and in situ hybridization to really understand how is it that these genes are changing in terms of their expression during development. And then finally, in the Solanaceae, we have one more tool, and that is the functional analysis by virus-induced gene silencing. So this technique relies on the cloning of specific sequences into viral vectors. These viral vectors are then transformed into E. coli, then transformed into agrobacterium, and then they get infiltrated into the plant tissue. Now, because these are viral sequences, they trigger in the plant an immune response. They trigger the activation of the RNAi silencing complex, and eventually this results in the downregulation of both the viral sequences as well as the target gene, resulting in what we know as co-suppression. So we have been able to standardize this virus-induced gene silencing technique in 3 different species with different fruit types. We have done so for Capsicum, annum, for Solanum lycopersicum, the microton variety, and for a dwarf tobacco, which is Nicotiana obtusifolia. And here I'm showing you the positive controls for this technique. Basically, what you see is that here we're targeting phytoendosaccharides. This is an enzyme that is very important in the chlorophyll synthesis. And basically, once that enzyme is downregulated, we can see photobleaching in those leaves as a result of the absence of the PDS activity. This was actually all done by a master's student who is now a graduate student in Valencia. Her name is Clara Ortiz, and I'll be telling you a little bit about her work as well. Okay, so I'm going to move into the results. And here I want to tell you that we were expecting that these genes were very different. And we have these expectations because we know that during angiosperm diversification, there had been a lot of whole genome duplication events that have been, in fact, well identified. So here I'm showing you a summary tree for angiosperm diversification, and the stars actually point to those different whole genome duplication events that have been identified. And basically what this says, what this tells us, is that if we take any model organism, take for instance Arabidopsis within the Brassicales, that genetic complement is never going to be easy to extrapolate, not in terms of the number of genes and not in terms of the gene function, to all other angiosperm groups. This is particularly true for those early diverging basal angiosperms, but is the same across angiosperms. No? For instance, take the case of the Solanaceae. The Solanaceae have undergone their own whole genome duplication events. So the genetic complements are not necessarily easy to extrapolate between different groups. So we were expecting that the genes involved in the genetic network for fruit patterning had changed between Solanaceae and Brassicales. Now, we don't start from scratch. We have been working for a long time in identifying the evolution of these gene lineages. So I'm not going to show you all the details, but we have worked on understanding how is it that these genes have changed across angiosperm evolution. And we have identified the evolution of the AP1 full gene lineage, the Agamus shatterproof, Aperla2, Alcatraz spatula, as well as the INDEHISCENT genes. And usually we start by doing this research, we start by identifying homologs in the public repositories, whether those are the genomes or transcriptomes, and even our own repositories. We have gathered around 80 transcriptomes from tropical plant species. This is done with the goal of really identifying where are those homologs well studied in Arabidopsis, and how does the genetic complement of Arabidopsis compare to other plant species. So let's have a look at the Solanaceae genes. So in the Solanaceae, there are some gene lineages that have not undergone any changes when compared to that network in Arabidopsis. There are few, but they do exist. So for example, the Agamus and shatterproof gene lineage underwent a single duplication in the chorioidicots, and it hasn't changed since then. So basically the same Agamus and shatterproof genes that Arabidopsis has is the same that both dry dehiscent fruited species and fleshy fruited species have in the Solanaceae. Same is true for the Alcatraz spatula gene lineage. A single duplication in the chorioidicots, the same genetic complement is retained in the dry fruited versus the fleshy fruited species in the Solanaceae. However, this is not always the case. We know that there are some fruit development genes that have undergone additional duplications. And this is the case of the Aperla1 fruitful gene lineage. There is a duplication that is shared with the Brassicaceae. So Arabidopsis and all the dry fruited and fleshy fruited species in Solanaceae have UFUL1 genes and UFUL2 genes. But in addition to those genes, the Solanaceae have undergone an additional duplication in each clade, resulting in at least 4 gene copies in that particular gene lineage. Same is true for Hecari. You can see how additional duplications in the Solanaceae have altered the genetic complement. Basically, the Solanaceae have 5 different Hecari copies, and they lack INDEHISCENT genes. So this is not unique for Solanaceae. In fact, INDEHISCENT is the result of a Brassicaceae specific duplication. So we know that INDEHISCENT genes don't have true orthologs outside of the Brassicales. And the same is true for Replumles. This is a very interesting case because Replumles is a gene that is mostly retained as single copy across angiosperms. And in the Solanaceae, we have an additional duplication and in turn, 2 copies present across species, Replumles 1 and Replumles 2. Okay, so what do we do with this information? No? If we already understand how the genetic complement has changed between Solanaceae and our model reference species Arabidopsis, then how do we go about studying these genes in different fruit types in the Solanaceae? So it seems that this might be an oversimplification, but it seems that if we're interested in understanding fruit diversity, the first genetic tier of the regulatory network might not be the one that to be looking at, because these genes start out their function very early on in development. So basically what I'm trying to tell you here is that if we know that the full shatterproof genes are super important since very early stages of fruit development. When they're downregulated in fleshy fruits, they affect dramatically the ripening processes. If full is overexpressed in capsules, the capsule doesn't open. If shatterproof is downregulated in capsules, the capsules don't release the seeds. So it seems that these genes are acting very early on in development and are not necessarily controlling different fruit types. The phenotype for fruitful is so well maintained in angiosperms that we know that these genes control very early processes even outside of the core eudicote. So the same is true for fruitful genes, even in basal eudicote. So we know these genes are basically important for the patterning of the pericarp, but are not controlling major changes between dry and fleshy fruits. So which genes to look at then? If we have this particular scenario, how do we choose some genes that we think might be more related to those subtle changes in the anatomy of the fruits? And something that we wanted to test with Clara and with Sayonara, the two students that were involved in this project, was which genes were actually changing a little bit more between the carpels, the early stages of fruit development and the late stages of fruit development from this genetic regulatory network that we could target as perhaps important for fruit patterning. And so here, two genes jumped out. Those are the ALCATRAZ spatula genes, and I'm going to tell you why. Mostly, we realized that spatula genes were maintained on in later stages of fruit development in the dry dehiscent fruits, and the opposite occurred for fleshy fruited species. You can see how spatula is turned off in the fleshy fruited species, but ALCATRAZ remains on the fleshy fruited species. So we thought, okay, let's try that. Let's look at the evolution of ALCATRAZ spatula genes. And let's try to see what we can find in terms of their expression and their function in different species. So we went ahead, and just as a reminder, ALCATRAZ spatula is a gene lineage that duplicated in the core eudicotes, which means that all core eudicotes, including Solanaceae, have both ALCATRAZ and spatula genes. Okay, so we started out by looking at the expression patterns of these genes. ALCATRAZ and spatula are in the dry fruited species Nicotiana obtusifolia, are not strongly expressed during flower development, but they are turned on later during fruit transition in the middle stages of fruit development. And something that is very interesting is that these genes are actually turned on in the endocarp. As you might recall, the endocarp is the layer that will become lignified. This is true for ALCATRAZ and is also true in situ hybridization for spatula genes. Now, something that it's very interesting is that if either gene is independently downregulated, we don't see any differences compared to the wild type phenotype. It takes both genes to be downregulated in order to see phenotypic changes. So in the double mutants, ALCATRAZ and spatula, you can see here the downregulation levels compared to the wild type gene expression. One can see that effectively the flowers are smaller and the fruits also prematurely dehisce. So they accumulate lignification, or delignification occurs faster. It occurs prematurely. And perhaps more interesting is the fact that additional layers of the endocarp become lignified in the double mutant compared to the wild type. So you can see in the wild type, there is 2 to 3 layers of lignified endocarp. And in the spatula ALCATRAZ mutants, there are 3 to 4 layers of the endocarp that become lignified. Okay, let's look at the fleshy fruited species. I'm showing you here results for Solanum, but it's the same for Capsicum, basically. The expression patterns of these genes in the fleshy fruited taxa start out early in development. So you can see that expression is active in the sepals, in the petals, in the stamens, in the carpels, as well as in the ovules, and even later at later stages in the stigma. And then something changes during fruit development, while ALCATRAZ stays on in the endocarp and in the pericarp, as well as in the ovules. Spatula genes are turned off during fruit development. Now, something that was very important and an observation that we saw in the, again, double silenced plants is that during flower development, you can see how this differs from the wild type phenotype in that the petals are not fully closed. So it seems that spatula and ALCATRAZ genes have even early roles in flower development. They might be important for the proper postgenital fusion of petals that protects the stamens and the carpels. And you can see how the two genes, when the two genes are missing, you can see how they prematurely expose the inner floral organs. Now, perhaps more interesting are the changes during fruit development. So you can see the double spatula ALCATRAZ mutants over here. In fact, the pericarp accumulates ectopic patches of what we think is lignified tissue. You can see that from the morphology point of view. And although it was very hard to get a good section, what Clara was able to observe is that despite the fact that the pericarp becomes a little bit mushy, the parenchyma starts to degrade, there are some patches of sclerified tissue in that pericarp when compared to the usually fully parenchymatic pericarp of the wild type capsicum. Okay, so basically, what does this mean for us? This means that spatula and ALCATRAZ genes are important, are likely involved in repressing lignification, both in time as well as in place, independently of the fruit type. So this is something very interesting because we saw changes in the dry dehiscent fruits of Nicotiana, but we're also seeing changes in the fleshy fruited species. This is something interesting as well. Natalie was able to observe that spatula ALCATRAZ as well as replumeless genes are perhaps the most interesting candidates for exploring convergent fruit types. So what she saw was that these genes are the ones that whose expression most likely or more closely resembles their phylogenetic position. So that's something very interesting as well. Okay, so I think that's all I wanted to show you, but I wanted to end up over here with this slide just to tell you that this is a work in progress. We have been able to identify how the genetic complements have changed in different species across Solanaceae. We have identified the number of copies that we from that genetic fruit regulatory network. And perhaps what we think is that it will be very important is to pursue now VIGs or functional analysis of those specific genes in the fleshy fruited species of Cestrum. No? That would be really, really interesting. We are hoping that by identifying not only these inside the Solanaceae, but also by including perhaps more genes and even more plant families into these studies, we can really assess at some point how is it that the fruit development genetic regulatory network has changed across angiosperms. So in order to do that, we're hoping to include eventually more plant families like those I already mentioned, and more genes, and perhaps understanding where those genes really came from. Many of these genes are angiosperm exclusive, but not all of them are. Okay, so this is my final slide. I would like to thank for this particular presentation. A lot of the early work that was done in the anatomy of the fruits was done with Rachel Meyer and Amy Leith when we were all at the New York Botanical Garden. Part of that research kind of became transferred to the Evo lab when I started as a PI in Colombia. And in this work, a lot of people have participated. So I am extremely thankful to Clara Ortiz, Sayonara, and Natalie, who are the ones that have been working on the Solanaceae fruit projects. And I'm particularly grateful with my international collaborators, Cristina Fernandez and Barbara Ambrose. Of course, all of the funding sources, we were very lucky and we had been lucky in getting some funds, both international and local funds. And I'm also grateful with my local collaborators as well, Fabio Gonzalez, Juan Fernando Alzate, and Marco Giraldo. This is our group. So this is an outdated slide already. There are some people here that have moved to do their PhDs in different places, but you can kind of see some of the people in our lab. This is the Plant Evo lab. In case you're interested in our projects, this is our webpage that was made by Sarita Muñoz. And here is a picture of us. This is my email. This is my Twitter account. And I will be happy to take your questions if there are any. And thanks for your attention. I'm going to stop sharing, I guess.
Okay. Thank you, Natalia, for that amazing presentation and for your beautiful slides too. And that is a very nice group from Divo there in Medellín. Now we have some space for questions. If someone wants to ask something, just turn on your microphone or your camera. I'm gonna read the chats in the meantime. Here is a comment from Franco that say that is an overwhelming amount of work and data. Congratulations to you, Natalia, and your team. It's a very interesting research, and fruits are amazing. Okay, we had a comment from Sandy Knapp. Great talk, Natalia. So nice to see how this has all progresses so far. Patricia Bedinger said that it's absolutely beautiful. Limbo said that was a great talk. Thank you for your talk.
Thank you.
And Richard Arms, they also say, thank you, Natalia, very nice talk. Lynn had a question. Lynn, can you turn on your microphone?
Hello, thank you. Wonderful talk, great data and great pictures, so thank you. I wonder if you could say anything about sclerosomes. Sometimes we call them stone cells in Solanum fruits. I don't understand them, so could you give us some insight into what they are and if they have a function at all?
Lynn, that's a great question. I don't think anyone really understands what they are. I wish I had kind of revised the literature a little bit for this talk, but from what I recall, sclerosomes are thought to be reminiscent of the sclerified endocarp. Phylogenetically speaking, they occupy the same position in the outer endocarp. So it seems that in that region, well, okay. So what's the endocarp and what's the mesocarp? No, I kind of didn't want to get into that detail because there is a lot of discussion about it. But basically, they are in the innermost portion of the fruit wall. They are never located, or from what I have seen in the literature, they are usually not located in the outermost region of the pericarp. And because of that structural position, they were thought to be kind of remnants of that sclerified endocarp. But I don't think they are. So when we do the sections, I haven't sectioned many fruits that have them. In fact, the only time that I encountered sclerosomes was in the Iochroma fruits. I know they have been recorded in Solanum species, many, I guess, but basically what they are is they are groups of sclerenchyma cells that don't really scatter around the pericarp, so that are retained together. And they might be more numerous than the seeds themselves. So from what we could see is sometimes they occupy so much of the pericarp that you see more sclerosomes than seeds in the inside of the fruits. When we saw the ALCATRAZ spatula mutant and we saw that patch of sclerenchymatic area, that was the first thing I thought, oh, there are some fleshy fruits that can have that, naturally speaking, those areas that are kind of ectopic areas of lignification. But I don't think we understand them, and I haven't observed that many to be able to tell you what is common for them and what might be more like autapomorphies for each species. I don't know if all sclerosomes are the same. I don't know if all form in the same way. So I don't think I have enough information to solve your question.
Thanks. It really helps a lot because as taxonomists, we struggle with whether those are taxonomically interesting or important to us. In some groups, it appears to be yes. I'm talking about Solanum mainly. But yeah, thank you.
I think that would be a character that will be worth studying, developmentally speaking, like getting species that you think perhaps have that feature fixed, and then really looking into the anatomy of the fruits and across a developmental timeline to really see if they change or they don't. And even perhaps compare those with closely related species that don't have them. That would be a really good project, but I don't have enough.
Yeah, thank you.
Mm-hmm.
Thank you, Lynn. And we have a question from David Jackson here, but I can see David. David, do you want to go ahead with your question?
Hey, yeah, thank you very much. And thank you, Natalia, for a beautiful seminar. I actually have a question. I'm interested in pumpkin fruit development, and especially, but related to your work. Is there much natural variation in thickness of the pericarp wall, and any genes known that affect that?
I think so, David. Thanks for the question. I am not particular, I don't follow particularly that literature, but I think there is a lot of information in Solanaceae about, especially about size of the fruit and form, so like shape. There are genes reported. I wouldn't be able to tell you, I don't have the names on the top of my head, but for sure, there are several loci that have been identified. And I think it's because they are so important in the domestication processes that really they have been like in the eye of researchers for a long time. So I'm sure that there are some, and in fact, I think those genes have been more worked on. They have more research than the structural genes. The ones that I mentioned today are, if you think about it, they're like the very early genetic regulatory network for really fine fruit patterning in the Arabidopsis network, but that doesn't mean that they are the key genes to change the fruit structurally speaking. So perhaps.
Yeah. I mean, I know the size genes from, obviously, Zach Lippman's work, but I'm thinking really about the wall of the pericarp, like the thickness of that.
There is one of the two mutants. I can't remember which one exactly. I don't know if it's the shatterproof homolog or the full homolog. I know that there is one that affects the number of layers of the pericarp. So in the mutant, you see a reduction of the layers. But I can't recall which one is it right now. I think it's a shatterproof.
Thanks. Okay.
Thank you, David. And Sandy's raising her hand. Please go ahead, Sandy.
Hi, Natalia. That was beautiful. That was absolutely fantastic. What I was going to ask you is so the sclerenchyma that's in the kind of capsular fruits is completely different to the sclerenchyma that's in these sclerosome bodies that are in the. So they're completely different origins. So what happens in things like Saraca and Lycium? Have you guys looked at Lycium?
No, Sandy, well, first, it's good to see you again.
Nice to see you.
Second, we never looked. We only looked at the things that we had at hand. And then when I got back to Colombia, I started to look mostly for gaps, major gaps. So we were especially concentrated on Cestrum and Brugmansia. So it's not like we have done an extensive sampling. I would love to be able to collect some more of those particular taxa that are, unique.
That are the kind of those that have funny and in between, aren't they?
Exactly. So none of we have not sampled any of those that you mentioned. I don't know if that's a good question about the sclerenchyma. I mean, if you think about cell-fate, sclerenchyma should be homologous. It shouldn't be that different. You would see differences between fibers and sclerates, but not really. I mean, the cell is just an sclerenchymatic cell. So the shape might be a little bit different in the sclerosomes. It's true. That's something that has been reported because it is a sclerenchymatic cell, but the shape is not, it's irregular. That's how it has been described. So basically it's not rounded. It doesn't become homogeneously lignified. It's almost like if it was a pavement cell that becomes lignified. So it is a little bit, so those are the ones that are different. I agree. So sclerosomes are perhaps not homologous to sclerates and not homologs to other early diverging sclerenchymatic endocarp. You're right.
Yeah, so they're interesting because they can be quite constant in number throughout a species. And then the shape depends on the packing of the seeds sometimes as well. So that this kind of packing, there's a packing thing even in those berry fruits, which changes the shapes of things, which is very odd. It's very peculiar, and we kind of use it taxonomically, but without really, as Lynn said, without really understanding what's going on.
I think that's something that really merits some detailed anatomical research. I bet that there are some common features as well that can be targeted, even used phylogenetically speaking.
There's German literature about sclerosomes from the 1920s, which is all in German. Really difficult, but really lovely. It was really nice, beautiful, beautiful talk. Thank you.
Thank you.
Okay, thank you, Sandy. We had a comment here in the chat from Janet Sullivan. She says that no wonder fruit types don't always fit the textbook description. And thanks for an interesting presentation, Natalia. Stacy Smith say, oh, Saraka, there will be a challenge. Even with a hammer, I couldn't get the seeds out. I know that Stacy is here. I want to add something. We had another comment from Rebecca that said that was a great talk. Thank you, Natalia, for the talk. And Stacy's here.
Well, yeah, I have wondered what the relationship is to those bones in the fruit and sclerosomes. I kind of think maybe they're different. I mean, it's not a little thing. I mean, the whole thing is full of bone. It's a very interesting and scary structure.
Does it dehisce, Stacy, at all or no? I mean, it's indehiscent. I've never seen it. I've only read about it. So
no, it's just a berry that's just full of just concrete inside. My conclusion is that they, they live in sort of boggy pyramidal kind of habitats. So I think that the fruits just fall and rot very, very slowly. But it would be, I would love to see someone work, on the sort of some kind of comparative analysis of all of these things. I've always been interested in sclerosomes, and I have also tried to figure out sort of if they had any phylogenetic structure. And it seems like within Iachromene, there's lots of things like this happening inside of the fruit flesh, but I'm really not sure how they're developmentally related. But Julia has a question that will be more astute than my blabbering about bones.
I don't know about that. I have a question on, unless I missed it, it seems like you focused, Natalia, most of your work on, for Brugmansia, on suaviolens. Did you look into other species as well, or no?
No, that's something that we also wanted to do, but we had to kind of concentrate our efforts in one. And we usually go to the easy one to collect. So there was one species that was growing in the Jardín Botánico de Medellín, and that's the one we took. So I don't even think it's a native one. I think this is all cultivated, and it's very hard to get the fruits from a lot of these Brugmansias. So to answer your question, we only have one species, but it would be great if we can do a more comparative analysis between closely related taxa. And even in Datureae, we see lots of variants in the shape and in the way they dehisce, but we only have one representative, which is Datureae inoxia. So, but if you have, I mean, if anyone in this audience is interested in pursuing some sort of comparative project in terms of anatomy, I would be more than happy to, collaborate, receive the sample section, kind of see what we see, and then let you know what we observe.
Oh, I ask because I did a part of my work during my PhD with 2 species of Brugmansia as well. We did some sections, but it wasn't on suaviolens. It was on sanguinea and versicolor. And they, as far as I remember, the fruits, they don't open. So I was surprised, actually. I contacted you during my PhD. You sent me that picture with the open fruit. So I was very surprised to see that. So I was wondering if you had a project going on the side for other Brugmansia species already or not.
No, not at this time, but the same picture that I sent you is the one that we published. I mean, it's very unusual. Sometimes what we see is that the pericarp kind of dries out, and then we only see the veins of the fruit really kind of surrounding the seeds. So Brugmansia is very, very unusual.
It's
And
very
I would
weird. I
love,
agree.
and I would love to have more samples across different species to really test what the variation is, especially because we don't even know what disperses those seeds.
Right.
So they at least the only ones that I have seen, they're always kind of rotting around, and the remnants are the vascular traces.
Right.
So I'm not
Yeah,
totally sure.
that's what I've seen also. Yeah, just wondering if you're following up on other species or not. Thank you.
But Julia, if you want to send those Brugmansias out to me, I will be.
So I do have some samples, but I don't have a developmental series. I have like some stages, but not a very well.
I think we just need to go out into the field and collect them again.
I think you're right. Thank you.
Thank you.
Thank you, Julia. I just had a comment related with Brugmansia. I know that the Perico Roda, working in the Universidad Nacional, they had access to all Brugmansia species, I guess. Maybe you need to collaborate with Brugmansia.
Maybe. And I think he might have developmental stages of more species. So that would be interesting.
Yeah, could be nice. Lina, do you want to ask something else?
Yeah, I just have another quick question. I think you said, Natalia, that Datura does not develop dehiscent zones in the fruit, and yet the fruit opens. But does it just kind of burst open? It just kind of explodes, but it doesn't have an organized way of opening?
So Lynn, I've seen both things. I was convinced that Datura, because I started to see Datura in the greenhouse at the beginning, I was convinced that it was only exploding irregularly. But then in the field, we see Daturas opening perfectly fine in a regular pattern, localicidally and septicidally. So I've seen both, but I bet that there are no dehiscent zones in so whatever is in those that are organized, that open in an organized manner, my prediction is that one wouldn't find a lignified endocarp or anything like that, because there isn't really, I mean, I haven't seen it in any of the Solanoideae members. It's not like I have studied them all, but I've never seen something like that. My prediction is that it has to do with the main vascular traces of the carpel and the way the tissue organizes around those vascular traces. Sometimes they can open irregularly, and I have seen Daturas that are perfectly opening localicidally and septicidally, but I have done just like quick sections in the field, and I bet you there is no lignified endocarp. My hypothesis is that the fibers in the spine, those spiny outgrowths, perhaps the fibers kind of serve as a mechanic tissue for tension, for producing the tension.
Okay, very interesting. Thanks.
Okay, I had a comment, more than a question, is a comment. I don't know that you see Nicandra pisalodes is quite a common species that maybe is at hand in Medellín. And this is a quite nice fleshy fruit, like a berry, a proper berry. But when it's completely ripe, it starts to dry, and that berry just release the seeds, just open it regularly and release the seeds. Do you have plans to study maybe Nicandra?
Is collected, Andres. Is waiting in the lab for us to get back and section it. And we have seen it splitting open from the bottom to the top, which is also unusual because it kind of breaks more easily in the bottom part of the fruit, which is something that I have not seen in other taxa. So thank you. But if you have other Nicandras in your collection and you want us to section that, please send them. But yes, it's in our list.
Now we have 3 species on Nicotiana. We need to talk with Peruvian collaborators to find the other 2. We had a couple of comments here. Stacy say that was so great. Natalia, thank you. Loretta Freitas says nice talk. Natalia, thank you. And Gustavo said I had a question. Gustavo, Are you there?
Yes, here. Hello. So thank you, Natalia, for this interesting seminar. I just want to know, maybe you can share some views at the microevolutionary level. If, for example, we could expect some differences in this regulation of the fruit development. For example, if I am thinking about some difference in this speed of the development following some environmental clues, if I will expect some fine tuning of these regulatory genes or regulatory networks, or maybe there could be like more differences in some gene duplications or something like that. Because I imagine that there could be like some moments that it's more convenient to release the seeds for some plants depending of the environmental differences surrounding the plants. So if there could be like some differences in different populations of the same species, I will expect some differences in these regulatory processes, or I will try to find some different genes, or what do you think about that?
Gustavo, I'm not totally sure. I think, okay, so perhaps this genetic regulatory network that I presented today, it's way too early for adaptation processes, because these are all structural genes. So for example, they wouldn't be changing in copy number. We know that they aren't. They are very stable across Solanaceae. Not when you compare them with the angiosperms, but in general terms, I think we only see duplications when it's a true polyploid and things like that. So these genes would definitely not be the right genes to be looking at for adaptation processes, I would say, mostly because these are very early structural patterning genes for pericarp morphology. For sure, there are others that are perhaps more environmentally that respond to changes in the environment, but it wouldn't be the structural genes. It would be perhaps later in the genetic cascade, something that is, I would imagine, it's not that I for sure know, but I would imagine that it would be genes that are downstream this genetic network. Perhaps, like you say, something that is related with the timing of the seed release, and that I don't know which would be. I mean, it might be just, we don't understand the timing processes here very well. We understand the changes, we understand almost when they start to occur in different fruit types, but we don't know if these features change in different species. So I guess let me rephrase my answer. Perhaps one way to start would be to look at exactly what are the differences between the species that are responsive or that respond to environmental changes, to really see which genes might be involved in those features. My guess is that these are way too early in the fruit patterning cascade, and that they would not be changing in terms of the environment. But I'm not 100% sure.
And just another curiosity, I guess a little bit more general, those genes related with flowering time have like some, like there are some genes both related in flowering time and fruit development, or like flower development and fruit development. There are some like same genes involved in that. Or?
So not really. So the gene families are gigantic. So for example, literally almost every process in plant development includes a Matchbox genes or a BHLH gene or a Hox gene, but different genes within those gene families. So there is no overlap that we know, or at least extensive overlap that we know between the flowering time genes and the floral organ identity genes and the fruit patterning genes, usually different genes, but same gene lineages sometimes.
Okay, thank you very much.
Thank you.
Thank Thank you, Gustavo. We had a comment here from David Esteban Granados that say, thank you, Natalia, for your talk. That was great. He said that he was looking for Brugmansias on the field, and some fruits had their ripened walls, and some of them are close to rivers. And he was thinking that maybe this is a form of dispersion. But there is a hypothesis that say that could be the disperser could be bats. I guess that is bats that he means. But the Brugmansia lost the dispersal animals. Maybe I'm using rivers to disperse or something.
That might be. That might be. I've never collected that many in the field to know, but if that's something very possible. Thanks for the comment.
Okay, okay, that's all in the chat. If someone else had a question here in the public, if someone want to ask something else. Okay, I think this is all for today. Thank you, Natalia, for your amazing talk today. For next week, on April 2nd, we are going to host to Carmen Fernandez and Víctor Quipuscoa from Facultad de Ciencias Biológicas de la Universidad Nacional de San Agustín de Arequipa in Peru, who is going to tell us about the interesting genus Leptoglossis. We had a final comment here from Thibault, Thibault, that said that was a beautiful word and talk, and thank you, merci. Thank you to everyone to be here today, and see you next week.